to review, we've talked about the cell wall structure in prokaryotic cells. We talked about the fact that bacteria have a cell wall made out of peptidoglycan. And we said that the archaea don't have this cell wall structure. Instead, they have what's called an S layer that replaces the cell wall, if you will. What it is a layer of material that sits outside of the membrane and helps protect this single-celled organism that we call archaea. Now, beyond the peptidoglycan cell wall in bacteria and the S layer in archaea, there are additional layers of material in certain prokaryotic organisms. And that's what I want us to talk about next. So let's start with the bacteria. They have a cell membrane, as all cells do. And then they have this cell wall made out of this very complex carbohydrate called peptidoglycan. Now, believe it or not, some bacterial species put an S layer outside of their cell wall. It seems very redundant, and in fact, it is redundant. You can imagine that from an evolutionary standpoint, a bacterial cell that has both a cell wall and an S layer must have had to adapt to very severe environmental conditions in order to require that sort of double layer of protection against the environment. Now, most bacteria do not double up like that. Most bacteria have just the cell wall outside of the membrane, followed by, again, some of these additional layers of material. And the most commonly encountered layer is called the glycocalyx. Now, there are two types of glycocalyx layers. One is called a capsule, and one is called a slime layer. And these are made out of protein and carbohydrate, just as we saw with the S layer, protein and carbohydrate. So this is a repeating theme that we will see as we examine structures that are on the outside of the cell membrane, outside of the cell wall. The glycocalyx varies depending upon the species of bacteria that you're examining. Some of them have capsule, some of them have slime layer, and some bacterial species have neither one. When you look at archaeal cells, they may have glycocalyx or they may not, just like the bacteria. Some of them have capsules, some of them have slime layers, some have neither one. Now, if we compare that with eukaryotic cells, we see very different materials. Remember, some eukaryotes are single-celled, some eukaryotes are microbes. There are other eukaryotes that are multicellular. So when we're thinking about, for example, the plants and the animals, plants have a cell wall. Outside of their membrane, they have a cell wall made out of cellulose. And you all know what cellulose is like. It's cellulose that gives plant material its rigidity. It's cellulose that makes a stalk of celery or a carrot have that lovely crunch in your salad. It's carbohydrate. It's a very complex carbohydrate. Animals are different. Multicelled animals are very different. They don't have cellulose around their cells. They don't have a cell wall. Now, when we look at single-celled microscopic eukaryotes, we see some important differences. They can have layers of material outside of their cell membrane. The fungi, for example, have a cell wall. It's not made out of peptidoglycan like the bacteria. It's not made out of cellulose like the plants. It's made out of another complex carbohydrate called chitin. And if you recall from our introductory lecture, chitin is not just found in fungi, it's also what makes up the rigid exoskeleton in insect species. If we look at the protists, the other single-celled eukaryotic organisms that we've talked about, remember that includes both algae and protozoa, our primitive animals, the protozoa. For algae, there is a cell wall, and sometimes it's made out of cellulose, just like the higher order plants. But algae can have cell walls made out of other materials. Remember, we mentioned agar, and we mentioned carrageenan. It depends on the species for algae, what their cell wall is going to be made out of. The protozoa, the primitive animals, don't have a cell wall, just like the more advanced animal species. One of the questions that I get from students when we talk about eukaryotic cells is where the organelles came from. Students often can grasp the idea that an organelle gives a cell the ability to compartmentalize different jobs. That makes sense. But I'm often asked, where did the organelles come from? How does a cell evolve to suddenly have these membrane-bound compartments inside of it? And the answer is, we don't know exactly where all of the organelles came from. But we do have a very good origin story for a couple of the organelles. And that is what we call the endosymbiotic theory. Both the chloroplasts and the mitochondria are specialized organelles that arose through evolution because of this process called endosymbiosis, and we have good evidence for this. So let's think about chloroplasts. Chloroplasts are an organelle that is involved in the process of photosynthesis. Chloroplasts actually don't have one membrane around them, they have two. And as I said a minute ago, chloroplasts are not alone in having a double membrane. Mitochondria also have a double membrane. Very strange, because other organelles just have a single membrane around them. The other really unusual thing about chloroplasts and mitochondria is that they have their own DNA. There's a small length of DNA found in chloroplasts and found in mitochondria that is separate from the DNA that's found in the nucleus of eukaryotic cells. And if you watch this DNA molecule through the life of the cell, it divides independently of the nucleus. It has its own life, it has its own existence. When you look at the genes that are in the DNA inside a chloroplast, for example, they're a limited number of genes, and they code for proteins that only the chloroplast uses. So it's almost like the chloroplast is its own little organism. And that's where the endosymbiotic theory comes in. We believe that both the chloroplast and the mitochondria were once free-living, single-celled organisms. They were essentially an early form of what we now call bacteria. So sort of a pre-prokaryote organism. Now, for the chloroplast specifically, because the chloroplast is involved in photosynthesis, the belief is that the chloroplast was once a free-living photosynthetic type of bacteria, primitive bacteria, and that what must have happened at some point in our evolutionary history is that a larger cell, a larger, very primitive cell, must have engulfed that photosynthetic cell and kept it inside. Again, this is what we call endosymbiosis. If you translate that word, endo means within, and symbiosis describes a relationship where both organisms benefit from that relationship. So imagine this larger cell coming along and essentially just sort of engulfing this smaller cell, taking it inside. This smaller cell was able to do photosynthesis. It was able to harness energy from the sun and use that energy to produce ATP. That larger cell would then benefit because it had a source of ATP inside of it now. The smaller cell also benefited from this relationship because the smaller cell was now protected, tucked away inside the larger cell. So there are two important pieces of evidence that we have for endosymbiotic theory. Number one is that the chloroplast and the mitochondria have a double membrane. Essentially, the outer membrane would have come from this larger cell, and the inner membrane would have just been the cell membrane that was around the smaller cell. And we'll take a look at a picture of this in just a minute. The second piece of evidence is the fact that both chloroplasts and mitochondria have their own DNA. That certainly suggests that they were once independent, free-living cells. On this slide, we can see a very simple drawing of the process of endosymbiosis, of a larger primitive cell engulfing a smaller primitive cell. And the artist has drawn in both what eventually became the mitochondria and what eventually became the chloroplast. So here's our larger primitive cell. All right, it's a eukaryotic cell. We know that because it's got this nucleus inside of it. So this would have been a very early, very primitive form of a eukaryotic cell. Here is a free-living organism that eventually became the mitochondria. And they've got it listed as an aerobic prokaryote because, of course, we know that mitochondria help cells perform this process called cellular respiration, and that's an aerobic process. That's a process in eukaryotes that requires oxygen. So here's this free-living prokaryotic ancestor, and here's our larger primitive eukaryotic cell. Now, this larger cell is engulfing the smaller cell. See how that's happening? The larger cell's membrane is sort of folding inwards and capturing that smaller cell in a little compartment, in a little bubble, if you will. Now what you have here is essentially a double membrane. The outer membrane came from the big cell, and the inner membrane was just the cell membrane from the smaller cell. This relationship must have been adaptive. It must have been helpful. It was a symbiosis for these two organisms. And that's why it persisted across time. That's why it worked. And that's why the mitochondria became an organelle. Up at the top of the drawing, you can see what represents another primitive prokaryotic cell. This one is capable of photosynthesis. Here you can see it being engulfed by the larger cell. Here it now sits inside the larger cell, again, with two membranes around it. The membrane that belonged to that smaller cell, and then the outer membrane coming from the engulfer. Again, this is called endosymbiotic theory. It's what we believe was the process that brought about both chloroplasts and mitochondria.